Hi, my name is Veronica Pshibillo. The date is November 5th, 2019, and I will be presenting on the determination of gender, genes versus hormones. So first, let's have the overview of chromosomes and organs for sex determination. So a couple terms we need to establish. External genitalia, that's all the external reproductive organs. And then the internal genitalia, that's the accessory glands and ducts that connect the gonads with the outside environment. And the gonads are the organs that produce gametes. So both females and males have them. The male gonads are the testes, which produce sperm, and the female gonads are the ovaries, which produce eggs. And so the female that produces eggs, the mother, and the male that produces the sperm come together, and the egg and the sperm are both haploid, and they come together to form a new individual that is a zygote, and it is diploid. And so both the mother and the father give 22 autosomal pairs and one pair of sex chromosomes to the new individual. And so the autosomes, they contribute to the directed development of the human body form and characteristics. So, for example, they help with the development of hair color and blood type. And then both the mother and the father also give one pair of sex chromosomes, which helps determine the sex of the new individual. So there are X and Y sex chromosomes, and an important note is that the X is larger, so Y lacks many genes that the X has. So if the new individual is a female, then it will have X chromosomes, and they will have one X from both the mother and the father. And so it is important to note that for females, two X chromosomes are needed for normal female reproductive function. And for males, if you have a male child, it will have XY chromosomes, and they'll have one X from the mother and a Y from the father. And the Y is important for male reproductive organ development. So I have a picture here that shows the parents, the female and the male. The female is XX and the male is XY. And so the eggs are haploid, and you can see how the eggs come together with the sperm to create a female versus a male, which will be a zygote, and it will be diploid. So next, the sexual differentiation occurs early in development. Differentiation does not occur until the 7th week of development. Before differentiation, the embryonic tissue is bipotential, which means that you cannot identify as male or female. So the bipotential gonad, it has an outer cortex and an inner medulla. So in the presence of the appropriate developmental signal, the medulla will develop into the testes. And in the absence of the appropriate developmental signal, the cortex will develop into the ovarian tissue. So the bipotential internal genitalia consists of the Wolffian ducts and the Mullerian ducts. And so the Wolffian ducts are derived from the embryonic kidney, and the Mullerian ducts, the U should have a little accent on it, but my computer couldn't figure out how to do it. So these ducts, depending on if the new individual will develop as a female or male, one duct will continue to develop while the other one will degenerate. And the bipotential external genitalia, that consists of the genital tubercle, the urethral folds, the urethral groove, and the labioscrotal swellings. And the external genitalia will develop into male or female reproductive structures. And so here we have a picture of the internal genitalia, and you can see how the bipotential gonad, so depending on if it develops as a male or a female, you will have, so in the male, the Wolffian duct continues to develop into the testes, and the Mullerian duct degenerates. And in the female, the Mullerian duct continues to develop into the ovaries, and the Wolffian duct degenerates. So then the male embryonic development, an important note is that the testicular development does not require male sex hormones such as testosterone. Testosterone cannot be secreted until gonads differentiate into testes. So the SRY gene, it is the sex-determining region of the Y chromosomes. And so in the presence of the SRY gene, the gonadal medulla will develop into the testes, and the gonadal cortex regresses. So in the presence of the SRY gene, a protein called TDF, which it stands for testes determining factor, will be produced, and it will bind to DNA and it will activate other genes. And those genes include SOX9, WT1, which is the Wilms tumor protein, and SF1, which is the steroidogenic factor. And these genes help with the direct development of gonadal medulla into testes. And so once the testes differentiate, 3 hormones are produced. So AMH, which is the anti-Mullerian hormone, which also has the name Mullerian inhibiting substance, so you may see it in both forms. It is a glycoprotein that is secreted by the testicular Sertoli cells, and it causes the embryonic Mullerian ducts to regress. And then we have the androgens, which include the testosterone and the dihydrotestosterone, which is DHT. And they are secreted by the testicular interstitial Leydig cells. And both androgens are dominant steroids in males, and they both bind to the same androgen receptor. However, they have different responses, as you can see. So testosterone, it converts Wolffian ducts into male accessory structures such as the epididymis, the vas deferens, and seminal vesicle. And later in development, it is also used to help control the migration of the testes from the abdomen into the scrotum, so later in development. And DHT is used for the differential of the external genitalia. So for the female embryonic development, it is like the opposite. So after development of the ovaries in female fetus, one of the X chromosomes in each cell of the body is inactivated, and it condenses into a Barr body. And a Barr body is a clump of nuclear X chromosome chromatin. And so either you'll have an active maternal X chromosome or an active paternal X chromosome, and it is random whichever one is selected. So in the absence of the SRY gene, and the presence of multiple female-specific genes, the gonadal cortex will develop into the ovaries, and the gonadal medulla regresses. So you see it's the opposite from male embryonic development. And so the absence of the testicular AMH will cause the Mullerian ducts to develop into the upper portion of the vagina, uterus, and fallopian tubes, and the absence of testosterone will cause the Wolffian ducts to degenerate, and the absence of DHT will cause the external genitalia to acquire female characteristics. And so here I have 2 pictures to show how the SRY gene controls the testicular and ovarian development. So the first picture is more complex, but it shows how the addition of the SRY causes the production of the 3 hormones we had discussed previously, and how it helps change the gonadal medulla into the testes, while the absence shows the hormones that are produced to help develop the ovaries from the gonadal cortex. And the second picture is a more basic picture, but it also shows which ducts degenerate with each development and what the ducts develop into. So you can see that the Wolffian ducts become the epididymis, and the ducts, the vas deferens, and the Mullerian ducts become the uterine tubes and the uterus. And here the Wolffian ducts degrade, and here the Mullerian ducts degrade. So it flip-flops. And I have included my sources. And yeah, have a good night.